Hey folks, if you need help on Tuesday's assignment, the guided reading, mitosis, and computer activity, you came to the right place. I'm also going to show you how to do Thursday's lab activity. So if you need help, here we go. All right, name, put your name on that. First name and at least a last initial so I can find you on Aries really easily. I'm going to go real fast, but guess what? You can push pause if I'm going too fast. Why is it important for cells to remain small? Because it is easier to move substances, in and out of the cell. 2, the relaxed form of DNA. This is when DNA is in interphase and you cannot see it under a microscope. That's when it's called chromatin. Now, when a cell goes into mitosis and it's going to divide, the chromatin squishes together, it condenses, and they form chromosomes. And that's when they become visible under the microscope. So you can see them with a microscope. 4, what are the 3 stages of the cell cycle? Cells can be in interphase, they can be in mitosis, or cytokinesis. 5, what happens? What is the cell doing in interphase? Well, the cell is growing. It's replicating DNA. So it's making a copy of its DNA. And it's just doing its normal job. So whatever that cell is, if a blood cell, its job is to carry oxygen. If it's a white blood cell, it's to fight pathogens like bacteria. What are the 4 stages of mitosis? Remember, that's PMAT. The P stands for prophase. The M stands for metaphase. A stands for anaphase. And the T stands for telophase. What function do centrioles play in mitosis? They move around the chromosomes during mitosis, which process pinches off the cytoplasm to form 2 new daughter cells. So what process actually turns one cell and it divides into 2? That's the last thing. It's called cytokinesis. How is cancer related to the cell cycle? Well, cancer is uncontrolled cell division. The cell just keeps dividing and dividing and dividing and dividing. It doesn't go into interphase. It just keeps making copies and copies, and it forms a tumor. That's what cancer is. What phases make up the cell cycle? We've already done it, but it's interphase, mitosis, and cytokinesis. When you make 2 daughter cells, how similar will the 2 cells, how similar will their DNA be? They're going to be exactly the same. Because when you make new cells, you want your new cells to be exactly the same as your old cells. You don't want your new skin cells to be purple or green or something. You want them to look like your old cells, so they got to have the same DNA. What happens before mitosis? Interphase. During interphase, inside the DNA inside the nucleus, if it's interphase, remember it's called chromatin. When it's uncoiled and stringy and you can't see it under a microscope. How many pieces of DNA, how many chromosomes are in your cells? Since you're a Homo sapien human, you have 46. What must happen to DNA before mitosis? It has to replicate. So it has to make a copy of itself. Why is it important to make new cells? Well, for things like growth, when you're growing, you need new cells to replace dead / damaged cells, etc., etc. What is the correct order of phases in mitosis? Remember, that's PMAT, so I'm just going to put PMAT. Prophase, P for prophase, M for metaphase, A for anaphase, T for telophase. What happens to chromatin during prophase? It condenses into chromosomes. So it kind of squeezes together, and the chromatin turns into chromosomes, and they become visible under the microscope. Each half of a chromosome, okay, so I'm going to draw it real quick. You do not have to do this drawing. But so a chromosome looks kind of like this. So each side of that chromosome, of that blue thing, that's an exact copy. Each side they're exactly the same. But each little leg, that little leg right there, and then that little leg right there, those are called sister chromatids. Sister chromatids. And once they separate from each other, so once one of this side or that side, once they leave, they're no longer chromatids. Now they'll be a single-stranded chromosome. What happens to the nuclear membrane during prophase? It disappears. What do the chromosomes do during metaphase? Remember, metaphase middle, metaphase middle. In metaphase, they line up in the middle of the cell. What happens to sister chromatids during anaphase? This is when the chromatids, they are pulled away from each other. Away starts with an A, so does anaphase. Same with the M, middle. Middle starts with an M, so does metaphase. So metaphase middle, anaphase, they move away. What's created during telophase? Telophase, the last phase, we get two nuclei. Nuclei is plural for nucleus. So you don't say nucleuses, you say nuclei. So we'll have two nuclei in one cell. Well, eventually that cell is going to divide into two cells, and then each one of those cells is going to have its own nucleus. And that process which divides one cell into two is called cytokinesis. All right, the drawing. I'm going to draw these really quick. Interphase. It's going to look kind of like this on today's lab. So drawing. All right, so the outside of the cell wall and the cell membrane. And then you're going to see a circle, and it's going to be totally shaded in. That's the nucleus. Totally shaded in. So that's what prophase is going to look like. The vast majority of the cells that you're going to see today are going to be in interphase. And then prophase, once again, the cell wall / cell membrane on the outside. Prophase is going to look kind of the same. It's still a circle, but now you're going to see little bars. So it's not going to be one, like an even color. It's going to be like you're going to see little bars like that. So that's what prophase is going to look like. Metaphase middle, remember? Metaphase middle. So there's your cell wall, cell membrane. And now the chromosomes are going to be lined up in the middle of the cell like that. Anaphase, cell wall, cell membrane. Boom, boom, boom. Anaphase, now the chromosomes are going to be moving away. So you're going to have a group of chromosomes over here moving to the left, and a group of chromosomes over here moving towards the right side of the cell. And the last one is telophase. You're going to see a large rectangle. And this group of chromosomes arrived. It's not going to be a circle, though. It's going to be kind of like an egg shape. There's going to be another one over here. New nuclear membrane is forming, and it's going to be colored in like that. All right, so then you might actually see the new cell wall / cell membrane forming in between. So we're going to get two cells right there. All right, so that's it for that. Now, today's lesson, let's look at it. First thing you're going to do is you want to go to Google. Well, first of all, boom, here it is. This is what you're going to see on Google Classroom. You're going to draw the phases again, but you're going to look at them. Now, what you're going to do is you're going to go to Google, and you're going to Google this, onion root tip mitosis. Onion root tip mitosis, you're going to get this. And pick this one right here. So it's like the third row down, boom. And then visit that page, boom, and then you can zoom in on it. But anyways, I zoomed in on this one. And what you're going to do is, so first, you're going to draw them. So I'm going to help you. So let's see. So the first one is interphase. The vast majority of these guys are going to be in interphase. So like this one right here. That's interphase, because you can see, it's a little too big. You can see the nucleus right here. There's the nucleus. The nucleus is still there. That's interphase. The next one will be prophase. Let's find a Here's prophase right here. Boom. See how it's still a circle, but now you can see the dark chromosomes in there. So that's prophase. Another prophase right here's another prophase. Metaphase. Here's a metaphase right here. Because you can see, look, they're lined up right in the middle in a line. Here's another metaphase up here. Anaphase. Here's an anaphase right here. Because look, these chromosomes over here are moving this way, and these guys over here are moving that way. Anaphase. And the last one is telophase or telophase. And there's one right here. Boom. And that's telophase. So you see a new nucleus forming right here. That is not a circle. Remember, it says it's going to be like an egg shape. And here's another nucleus going to be formed here. Eventually, these are going to split in two. All right, so now this one, I'm going to help you get started on this one. You can go like this, first of all. You can do this, to the last 3 columns. So interphase, you got to count how many cells are in interphase. I'm going back. I'm going to count them. All right, there's 1, 2, 3, 4, 5, 6, 7. That's prophase. 8, 9, 10, 11, 12, 13, 14. That's prophase. 15, 16, 17, 18, 19, 20. That's prophase. 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32. I don't know what that is. 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45. That's anaphase, prophase. 45, 46. I don't know what this is. 47, 48, 4. That's prophase. Oh, we'll go this one. 49. So you're going to go here. And you're going to say text box, hey, how many were in interphase? 49. And then you're going to count all the prophases. So you find the prophases. So here's one right here. So you would count that guy. Count them all. 1. And there's 2. And just keep counting all the prophases. Put them in there. Count the metaphases, anaphases, telophases. And then do your best on these questions. And if you have any questions, we can take care of it next week. All right, so that's it. Try to get this done because this does not take very long at all. So I will see you guys next week.